Hello, everyone. I am Eric Gathirwa Kariuki, and I'm delighted to be presenting our work on the metatranscriptomic analysis of the gut microbiome of black soldier fly larvae reared on lignocellulose-rich fiber diets that unveiled key lignocellulolytic enzymes. This work was published in Frontiers in Microbiology Journal in April 2023. Let's get started. Second generation biofuels, also known as 2G, from lignocellulosic biomass have emerged as a sustainable energy option to fossil fuels. However, production obstacles hinder the achievement of optimal performance in the 2G bioconversion process under minimal capital investment. Efficient production of 2G biofuels requires affordable and time-efficient pretreatment and hydrolysis methods, processes that are typically aided by lignocellulolytic microbes and enzymes. The black soldier fly larvae are known to degrade and valorize a wide range of organic biomass, and this is attributed to their potent gut microbiome. It is important to note that the black soldier fly larvae is characterized by 5 larval instars, a prepupal, pupal, and adult phase. All feeding is, however, restricted to the 5 larval instars as the fly does not feed as an adult. This, coupled with their rapid growth and inability to transmit any known diseases, present the larvae as a promising source of lignocellulolytic microbes and enzymes. Such microorganisms have the potential to be utilized in the production of different products such as biofuels and biorenewables. There are different ways that are commonly used to study complex microbial communities. We have targeted methods such as the 16S rRNA gene profiling for bacteria and archaea. Then we have shotgun methods that sample the entire genetic material across a community. For example, shotgun metagenomics uses DNA, viromics uses viral particles, and metatranscriptomics, which was used in this study, utilizes mRNA sequences to decode the active functions of microbes in their natural habitats, revealing their specific contributions within that community. It was used in this study to provide insights of bacteria with active functions in the larval guts at a higher resolution. The objectives of this study were to determine whether dietary intervention significantly alters the black soldier fly gut microbiome profiles, and then to design and implement a metatranscriptomic analysis pipeline to profile microbes and enzymes associated with lignocellulolytic functions in the larvae. From the inductive coupled plasma mass spectrometry analysis carried out to determine the fiber content of the diets that were used to rear the larvae, processed chicken feed had the lowest fiber and was used as the control diet. The experimental diets were ordered in increasing fiber level as follows: chicken manure, brewer spent grain, and water hyacinth. After rearing the black soldier fly larvae on the different diets, guts from the larvae were aseptically dissected before RNA extraction and mRNA enrichment to remove ribosomal RNA contaminants. After the preparation of sequencing libraries, sequencing was performed using the MinION platform, and the reads were bisculled and demultiplexed using GAPI. Quality control was then performed to remove unclassified reads and adapter sequences before clustering the transcript isoforms from the same gene using ISONclast and correcting them using ISONcorrect. The corrected reads were then mapped to the black soldier fly reference using the Minimap2 splice-aware aligner, and the unmapped reads were obtained using some tools for annotation against the RefSeq and SID subsystems databases with the Diamond aligner. Differential expression analysis was conducted in DSEQ2, and the most abundant genera were then identified in each metatranscriptome, followed by annotation of carbohydrate active enzymes with dbcan2 and the CASI database. From our results, we observed that the highest dissimilarity between metatranscriptomes was observed between the control samples on the left and the fiber-rich water hyacinth samples on the right, as illustrated by this Euclidean distance heat map. The metabolism of predominant bacteria in the pooled metatranscriptomes also showed that as the lignocellulose content in the diet increased, there was a reduction in the aerobicity of the gut. Therefore, more anaerobic and microaerobic bacteria species were observed in the guts of the larvae that were reared on the more fiber-rich diets. The relative activity plots showed distinct microbial profiling between the control metatranscriptomes on the left and the experimental ones on the right. Notably, the facultatively anaerobic Dysgonomonas species with widely reported lignocellulolytic functions exhibited notable activity in the guts of the larvae that were reared on high fiber diets. This observation was, however, absent in the control samples where bacterial genera such as Fingobacteria, Wixella, and Myroides were more dominant. Additionally, the screening of carbohydrate active enzymes in the dominant bacterial genera per diet revealed alpha L arabinofuranoside classes, glycoside hydrolase 43 and 51 exclusively in the guts of the larvae that were raised on fiber-rich substrates, that is brewer's spent green and water hyacinth. Furthermore, the larvae raised on these high fiber substrates had in common two bacteria species, that is Dysgonomonas and Bacteroides, which are well known for their widely reported lignocellulolytic capabilities. Besides Dysgonomonas and Bacteroides, other species such as Parabacteroides and Prevotella with reported lignocellulolytic abilities were identified in the guts of the larvae, also reared on these high fiber diets. The higher incidence of anaerobic bacteria in the guts of the larvae reared on high fiber diets is suggestive of lignocellulolytic activity that is reported to occur in the mid and hindgut sections in lignocellulose degrading insects. The joint catalytic action of alpha L arabinofuranoside cazymes, that is GH43 and 51, that were identified in the high fiber metatranscriptomes, is capable of the complete breakdown of the hemicellulose fractions in lignocellulosic biomass. This renders them potentially useful in the valorization of the less utilized hemicellulose in biotechnological applications. Therefore, metatranscriptomic analysis shows immense potential as the first line of bioprospecting microbes and enzymes with potential biotechnological applications in complex microbial communities at a higher resolution compared to classical microbiome profiling methods. If applied, this could potentially help in lowering the production costs, increasing the bioconversion efficiency in biofuels and other biorenewables. Here are a few useful links, such as the link to our paper and the GitHub repository where the scripts and the analyses generated for this study are domiciled. I would like to acknowledge the sponsors and facilitators of this study, as well as my supervisors and colleagues for their invaluable support, which played a crucial role in the successful completion of this work. Thank you.